Hi everyone, welcome to chapters 14, 15, and 16 as we kind of go over the kind of the question of DNA versus RNA, and this is something that's going to come up specifically in the lab that we're doing too, since it's a very big question in a lot of diseases and organisms, things like that. The DNA has kind of been kind of the crown prince as far as, what genetics is comparatively to RNA for a long time after the genome. And we kind of talked about how RNA only made a real comeback as, a molecule that was really studied after the HIV genome was, found to be RNA. So in this case, we're going to take a look at some of the complex machinery that goes into expression of genetic material. And that's going to bring up some exciting questions about what some of this machinery actually is. And that's going to bring us to kind of try and consider viruses as, something a little, they're not just disease-causing things. They are genetics, kind of come to life, if you want to put it that way. Obviously, you can definitely disagree that viruses are alive. There's a lot of evidence that would point that, these are not living things, but it's very exciting to look at, kind of how they play into this idea behind gene expression, gene manipulation, things like that. So chapter 14 is chapter 15 is the main thing we're covering because this is the most important stuff. 14 and 16 are kind of going to bookend things, and they're more important to understand some of the importances beside, kind of outside 15 that will kind of supplement your understanding of 15. Okay, so this is crossing over. Remember, this is when, on the nice little figures, usually the DNA just switches and it's fine, but it is a very complex process and it is facilitated so that this DNA is going to interact and basically switch and integrate into the other chromosome. So a good example of this, and I want you to consider, this is something we consider pretty normal, but what is the actual machinery? What does that actually mimic? Putting DNA and integrating DNA, switching, cutting and pasting DNA. Where does that come from? And a lot of the machinery in our cells is hypothesized to become or come from viruses. And come from viruses, I mean like back in the eukaryotic single cell development style of stage, and likely that these machinery, precursors were viruses infecting these protocells. So as you can see here, there's all kinds of, unique ways that viruses can get in and send in DNA. Some of them are found in nature, DNA and RNA viruses, like our friend coronavirus here. Some are nanotechnology, where we surround viruses with genetic material we want to deliver in a layer of liposomes, or in a liposome, for example, that it's going to integrate with this phospholipid bilayer right here. Cationic polymers, these are ions that are going to get to pass through here, send in their DNA, and then dendrimers are really interesting. They're just these chemical shields around genetic material to deliver DNA. So these three right here, these are nanotechnology, for example. This one over here too. Anything that you can deliver genetic material into. We often use a virus, and this is kind of what gene therapy is, and we saw this in one of the journal clubs. But for the most part, we're going to see that a lot of these natural mechanisms, especially over here, that integrate DNA, things like that, see how this is going to put DNA in. That means chopping DNA, cutting and pasting, everything that we see up here, for example. So there's a lot of schools of thought that say, okay, all the cool recombination machinery, a lot of the DNA editing stuff, all of that, like chopping, enzymatic activity, things like that to rework our genome, comes from precursors that were viruses that infected the genome, but they were reutilized and they gave an advantage to those cells that reutilized them in a beneficial way. So let's take a look at three examples. You can look in your book for more details on these. Three examples of how the genetic material can be changed and manipulated physically, and how this is going to relate to those transposons we've talked about, and how this kind of makes the genome, again, this very dynamic thing. So again, we've kind of covered the idea. Obviously, you have genes, you have sequences that promote the gene, and those that stop it. One way to turn on a gene is to literally take out the stop operator. Remember how the lac repressor is going to bind to this sequence? It can't bind and stop this gene from coming on if it's literally gone, right? And that's exciting, and that's saying that there's an enzyme that's going to pull this out, and one of these to pull out here too. So that's a very cool, machinery. Again, the idea that you can pull sequences of DNA out, and that's going to allow transcription. So remember, get the idea of transposons, those jumping genes in your head. They can also be removed through certain targeted mechanisms, and this is going to be a very cool way to activate genes like this. Okay, another way, this is kind of another simple idea. Remember transposons, the jumping gene? This is the same thing, is that they could take a gene, for example, from somewhere else, and they're going to put it into the genome and have it start expressing. What we're seeing right here is that this is a circular genome. This is a plasmid in this case, so this can be linear or it can be a plasmid like this. We're kind of working in a bacterial model here. But again, this is going to add to that kind of that conversation about how prokaryotes are so much quicker at adapting and introducing new genetic material. Even though they go through binary fission and they clone themselves, you can introduce genes so much easier just like this. So it's a very cool idea that a gene itself can be just inserted and you have a new gene. Another very cool thing, and this is where we start getting into eukaryotes, is allele replacement. So this isn't purely eukaryotic, but as you can see, this is starting to mimic that idea of crossing over, where you have similar alleles, but they're a little different, as you can tell right here, this little apostrophe. This is the beginning of the idea of, okay, crossing over and that holiday junction, everything that we see that gives like the wild variation that parents pass down to, their offspring, that has roots in viral mechanisms that cut and paste or chop DNA and integrate and reintegrate it. Because you can see right here, on this chromosome, you had this initial purple allele. You can end with something totally different. Well, in this case, a homolog. But still pretty close. So it's pretty awesome. So this is just an awesome picture in your book. This is using a Cree recombinase sleeping beauty system. I'll let you read about that if you want. Those of you doing grants on inducible systems, I'd read up on that because it's a very good system that you could utilize. In this case, different neurons were given kind of the same viral machinery, but the Cree recombinase reacted differently to activate different color genes, and thus you can see all these neurons start expressing GFP, RFP, YFP, BFP. So I don't actually know if BFP is the actual name for that, but still, I know GFP is yellow, red, RFP, yellow as YFP. But it's really interesting that you can send in genes and activate them, and that this activation can kind of show that the disposition towards that machinery. So it's kind of cool. Okay, transposons themselves, these are kind of the strict ways that you do things. There are genes that are going to make, and remember, this gene right here is actually going to make a protein that is going to excise itself from this portion right here and here. So those are very specific genes in that case. And it's going to then reinsert itself into a site that is going to match its enzymes, kind of its target site. And these nice reactive OHs are going to make that a lot easier. They're going to insert. Your DNA mechanisms will just repair that nicely. And you have cut and paste into a new chromosome. In this case, this is your copy and paste. In this case, you make a copy, you cleave, you find it, and in this case, the chromosomes will come together in order to facilitate kind of a junction right here, kind of a holiday junction, but I think that's fairly specific to crossing over. And in this case, it's going to replicate itself into this new section. So here, this cleavage is incomplete. It's going to cause nicks that allow this reactive OH group to come out, and that's what's going to kind of come into this junction here. So that's a really cool mechanism. And again, remember that these transposons, these are old viruses. These are jumping viruses that integrate their DNA and their genetic material, okay? Just like we see with retroviruses in a lot of cases. This is a very interesting format. This is one I actually haven't studied as much. They actually do make an RNA copy. So they have DNA, so they have themselves hidden in the DNA. They make an RNA, like, kind of like a reverse, or actually kind of like a normal gene. So you transcribe DNA to RNA. This gets reverse transcribed into a new fresh piece of DNA right here. That's put in. You cleave a little bit to add these nice OHs, and again, you kind of follow the same protocol as over here. You insert somewhere new in the genome. And so having that RNA intermediate, you also are going to require reverse transcription, though, in that case. So these are three really interesting ways that DNA and other genetic material can kind of jump. So I kind of talked about this. These genes don't just code, for nothing. They code for these proteins up here, that the whole role of the jumping genes is to make these large protein complexes you see here in gray and tan, that these transposases and these integrases, that their whole active site and their material is going to find specific spots in DNA and, basically cleave or insert into new target sites. And again, they usually do have a target site. I think some transposons are fairly random, but at the end of the day, I think we have a pretty good consensus of what they actually jump into. So in this case, that's the whole role of this DNA, is to make proteins that jump the DNA around, and that's very interesting. This is where we get strictly more into a disease where you have a nice reverse transcriptase protein ready to go, you have RNA, the RNA goes in, the reverse transcription makes the viral DNA, and then that integration happens right here, and now it's just part of your genome forever. That's why this is such a good tool. For example, when we use this in research, so a lot of you are inserting genes, retroviral integration is a great idea. You would have to look up what some of the weaknesses are here and kind of account for those, but otherwise, it's obviously something that we use quite a bit. So in this case, this kind of finishes out the viral life cycle, and it goes up and makes more retroviruses. So as we're going through this lecture, kind of keep in mind where the genetic machinery that we kind of take for granted, as it moves and, recombines our genome, how that usually behaves maybe in a system of disease like we see here. In this case, it's going to make a product that is going to keep propagating this product, and that's obviously not great, because in order to do that, oftentimes you have to blow the cell up through lysis. So throughout all this process, keep in mind that machinery is very, it's a very delicate balance between disease and machinery in a lot of these cells. So keep that in mind when we kind of look at the virome. So one of the best ways that we actually utilize a lot of these recombining jumping gene type stuff is making the antibody. So remember, these little sites up here on this large Y-shaped protein chain, the antibody, these are what bind the antigen sites. We know that in this variable region, these tips up here, that's where that somatic hypermutation shows up. But one of the other interesting things is that there are variable regions of these genes. So there are V genes, and they go, and they're all in the same, they're usually, I think they're all in chromosome 6 for the most part. They're all next to each other, and you basically get to choose one to make your gene out of. So you kind of get to choose different full genetic segments that you make your antibodies out of, and the decision to cut and paste this into the right spot and decide which one, again, that's like kind of an old viral mechanism. So it's very cool that to diversify the structure of these Y-shaped antibodies, you actually choose different segments of genes, and they cut and paste into the right site, and they all become a single gene that is going to create this nice antibody. So you have to choose, for example, you have to choose a large J region, you have to choose a large D region, you have to choose a large V region. And in that case, that adds to the diversity that you're already going to be, hypermutating up here in these red regions, so that you bind the antibody the best, or the antigen the best, because remember, that's what you want to bind to get rid of your pathogen. So very cool stuff. Again, this is kind of my field of research, so definitely let me know if you want to talk about this. It's awesome. All right. So one last really cool example of human evolution and the kind of the necessity of machinery that usually doesn't need to exist. So why is the Y chromosome this tiny, terrible speck of genetic material? Like we've talked about, there's about 45 to 60 genes on this thing. It doesn't add a lot. The X chromosome is really large. It has a ton of important genes on it. What happens? And this is, remember, this is all starting in like kind of hypothesis land, is that, the evolution of sex is still something that's very hotly debated in how this happened. The idea is that, so we know that there's a gene called SRY that is the triggering mechanism in development that will trigger the male phenotype. One of the prevailing hypotheses is that the SRY mutation caused it to become kind of this master regulator. At that point, and for whatever reason, the evolution of sex was, a positive in this case, you could basically lose, over time, you could basically lose anything on this Y chromosome as long as it wasn't the SRY. So what we see now today over here, and after, years of damage to the Y, it's this like really small, kind of pathetic little chromosome. It's not that great anymore, because every time the Y has a mistake or a deletion, but it doesn't target SRY, it just goes on. It just passes on again, because there's nothing in the phenotype that allows it to repair, because it doesn't have another chromosome. You don't have two Y chromosomes, you only have one. So in females, 2 X's means you can cross over, you can do recombination, and that means that if you're stuck with one bad allele, you don't necessarily always pass that on. With a Y, you can't really recombine with any of these genes. They don't have any copies or backup copies that you can have in case one gets mutated. So if you lose or get deletions, the Y basically, shrinks into this tiny thing here, and it's not very good. But how is that possible? How does the Y actually at least maintain some genetic integrity? And so remember how, the mom and the dad chromosome, they cross over, they recombine, they can kind of repair themselves like that. The Y can't do that with another partner. And sorry, this is such a terrible image, but it's the one that I wanted to use. The Y is made up of copies, palindromes of genetic material, and itself recombines. It switches right here with gene homologs, and that recombination can occur like that. It's, I think, the only chromosome that does this. It is intrachromosomal recombination. So this is very cool. And what it is essentially is that there are copies of genes on the Y. And if you lose these genes, you do have a backup system, kind of a backup copy that you can kind of use the right one for. Now, again, the other reason that the Y basically survives and doesn't just degrade is that if you lose one of the key genes on the Y, that usually leads to male infertility. You don't pass on the genes. That disease phenotype never passes on. That's kind of the unique thing about any condition that causes infertility is that, it's not going to be something that spreads because inherently it can't spread anymore. So this is a very cool mechanism, and a lot of this machinery that causes this, again, is viral, at least in origin. Okay. So cool. On to some of the kind of more concrete concepts. As we debate and kind of look at DNA versus RNA, we want to take a look and see if this molecule is something that represents a more dynamic picture of the genome or the organism or the DNA than does DNA blueprint. So in this case, this is just a great figure of how this DNA is going to open up. You are going to copy the information from the nice safe blueprint and turn it into an RNA. This RNA will go on to be translated to protein. The RNA polymerase has a pretty similar, kind of phenotype, chemical phenotype that the DNA polymerase does. Again, it's going to rely on ions to ensure that as it is adding in that 5' to 3' direction, they are going to basically bind up these incoming little RNAs, bind up here to match. And remember, this is just a match. They're not actually going to bind here. This is the bind that you're going to make right here, is this O. As you can see, it's jumping and attacking this incoming adenosine right here. So again, it's kind of like DNA replication. You're going to be adding nucleotide after nucleotide, except this strand is going to break right here, and this DNA is just going to reanneal after you've activated this gene. Okay. This is very cool. Again, this kind of highlights the cool, kind of applications that we saw from those topoisomerases. In this case, this is overwound DNA. It's positive supercoils. These are negative supercoils. Again, all those topoisomerases are very important when it comes to relaxing this DNA and making sure this doesn't literally just crack under the pressure, because you are basically twisting this DNA to facilitate this cool intermediate right here within the RNA transcription, the RNA polymerase. So it's a very cool thing that this basically winds through DNA and produces a nice RNA product, and it's all this, this great complex of enzymes. It's really cool. Okay. So there's plenty of things that we can use to study this, and anything, though, that stops RNA transcription is basically a full organism poison. Think about it, right? If you can stop and intercolate DNA like this acridine does right here, and you basically stop RNA polymerase from ever acting on this section, that gene will never react. So this is terrible, because if you spread this throughout your genome, you'll have tons of spots that, you can't make genes anymore. You just destroyed a bunch of genes if you can't unzip the DNA. That's why certain poisons, things like this, are really terrible. One application to medicine of this, though, is, we talk a lot about how, let's say we want to shut down a gene. If we could target what genes this actually inhibits, that would be awesome, because that'd be a really good system to get rid of genes in a living organism that, we don't want on, let's say for a disease or something. And we could basically just bind up that region of DNA. Now, again, this also likely causes some problems with DNA replication, as you can imagine, but it's not the easiest thing. Again, this is just another cool example. This mushroom makes this alpha-amatin. This basically goes in and ruins your RNA polymerase. It just binds up the middle of it. So if you have amatin, see right here, it is going to basically ruin polymerase, the RNA polymerase right there. You don't have any pre-mRNA coming out in this case. So you still have your ribosomal RNA and your tRNAs getting made, but nothing from your genome from pol 2 is going to get made from RNA. So kind of a bad deal. Don't eat mushrooms from the forest. Okay, so we've talked a ton about epigenetics, but this is kind of the big, the big details in how this starts. Remember, we've done a lot of work on histones and opening up that promoter. We're here at the promoter now. And I mean, given the class circumstances, I think we'd cover a lot of the stuff like more strictly in class, but obviously just given the circumstances is tough. But there is a whole nother world to, gene activation after you get this DNA out, like we have here. So as you can see here, this DNA is out of the histone, right? The RNA polymerase itself is going to bind to specific regions, usually a TA box, right, like right here, see that? And then there is going to be another kind of promoter spacer region right here. These are basically the active binding sites of an RNA polymerase. They need these sites around to find the gene that they're going to basically turn on. You need these regions active. You can also really manipulate the amount of RNA transcription, for example, by manipulating these regions. So this is actually a really good figure. If you're looking at, the wild type lac promoter up here, here's the wild type up here. Here's the typical sequence. If you don't have a promoter, obviously you have very little transcription because nothing's really going to bind there ever. So with the normal promoter, it's like kind of on, but if you manipulate and look in white here, you can see the different changes that you make in mutants. In this mutant, you have a deletion right here, for example. That goes up a little bit. See how that kind of goes up just a little bit, not down. See how this is kind of going to go up a little bit. Mutant number 2 goes up. See how maybe this makes it more attractive to the RNA polymerase. Mutant 3 goes up again. This is going to have the deletion and this double A. But mutant 4 goes down a little bit because you introduce this G right here. So kind of measure this sheet. You can go back to when I didn't mark it up if you want, and kind of see that even these small nucleotide changes can be something that we measure, because here's just the straight up gene activity. So you can measure gene activity and kind of the results of your mutations, your epigenetic mutations in this case. Because remember, this promoter, the promoter region, is not coding. So this is an epigenetic, these are epigenetic mutations we're looking at here. So this is really interesting because this shows you how gene dosage can be modulated by small genetic changes. Isn't that cool? This is the whole idea behind, small changes in amount of a protein. This is how you have different personalities, different variations. Not everything is so black and white with epigenetics as it is with Mendelian DNA in a lot of cases. So this is very cool. Okay. RNA polymerase is going to, like I said, kind of add this way. Here we go, hooray. There are two main hypotheses for proofreading. One is kinetic, where, as you can see, this U is trying to bind to a C. That's not really going to work, and so it's going to cause that bulge and not really be super happy with that. And it's going to push it back and redo that. So this, sorry, I should have mentioned, RNA polymerase does have fidelity mechanisms like the DNA polymerase, but it's far less accurate. You don't really need to care about the RNA that you're spitting out over and over. If you have a bad RNA, it might make a bad protein one time out of 10 million times. It's not a huge deal. I mean, the math may be a little wrong on that, but replicating DNA, that's permanent. That blueprint goes on to new cells. This RNA only exists in this cell. So that's why it's very, it can have a little more freedom and the speed can be a little higher. In this case, this is another hypothesis, nucleolytic proofreading. If it notices that there's a mistake, it can actually just remove entire sections like we see here and just start over after that. So it has good exonuclease possibilities. It has the kinetic possibility to basically detect when you've made a mistake. So again, important but not super important, not as important as DNA. Okay, there's two main termination models. So when you reach the end of a gene, in a lot of cases, the RNA that you're spitting out here, see how you're kind of growing this RNA strand, eventually there is a sequence that is going to form that hairpin loop, one of those wild structures, and the hairpin is essentially what breaks the RNA strand free of the RNA polymerase. So that's one hypothesis. The other is that there are DNA or RNA sites called rut sites that basically show up, and a helicase is actually going to bind those. And upon that site being bound, the helicase is going to keep moving and winding like we've seen helicase do. And the helicase is actually going to be the one that splits the RNA off of the RNA polymerase in that case. So 2 possible models, both likely have a role in some measure. Okay, this is an important slide because this is something that I don't want anybody to like have wrong or not consider in their grants. Everybody's grants is dealing a little bit in gene expression of some kind, so make sure that you have all these ideas, well, and what each one does. So in this case, you have your promoter, you have your gene. As long as RNA polymerase is going to find this and bind this, you're going to activate this gene. To get to that point, what you actually have to do is a lot of transcription factors, so a collection of different proteins are going to find the promoter. Dang it. They're going to find the promoter and bind, basically. Okay? That's what's going to attract RNA polymerase. Then we can get going on the gene. So obviously this isn't always a black and white, ingredient set, but those are the basic things. The other is that there are regions of DNA that are not near the promoter or the gene. Usually they're on the same chromosome like you see here, but they can be on different chromosomes called enhancers. Enhancer regions of DNA, these bind activator proteins, so these are different than transcription factors. These bind activators, and the activators in a lot of cases with genes, these are what are going to attract the transcription factors. So we kind of went in reverse here. So to get all the ingredients together, what you'd first need, let's say, is activator proteins need to bind their enhancer. This is going to cause the enhancer to kind of bend, and we'll see that there are models and there are proteins that do that bending for us, and it's really cool. These activators now position and make the perfect kind of, this perfect little environment right here, this perfect distance for transcription factors to bind each other, bind the activators, and then bind the promoter. And then at this point, RNA polymerase, once it sees the transcription factors, it has the active site to bind those, and then it's going to bind the promoter. We activate the gene. These are the main things to consider across any of your grants, everything like that. Don't leave stuff like this out, or not consider some of these ideas, because that is how you basically activate a gene. In this case, you'll see that, and especially in your pitfalls section, not considering these epigenetic ideas can lead to kind of disastrous research plans sometimes. Okay, this is a large eukaryotic representation of this. Again, eukaryotes are going to need a lot more factors, so these transcription factors up here, these are kind of placeholder genes. These genes actually exist, but these aren't always the same ones that bind, for example, the RNA polymerase. So in this case, you can see this enhancer DNA and basically these activators right here, they're binding up, they're going to attract this large mediator protein that is going to facilitate a perfect area not only for all the transcription factors, but you can see right here the RNA polymerase itself. So this also plays in that eukaryotic gene expression is very complicated. There's a lot of different steps. You have a lot more, this allows you more opportunities for gene dosage, a lot wider range and control over how much gene is made, but you are far less responsive to any change at a cell level, the way bacteria can just activate genes once it needs them. And if you lose one of these genes, all of a sudden you are messing with an entire system and multiple genes that you are regulating. So it's really tough in that case. Okay, so we're just at regular RNA. This is just, I should have had this up as we were talking about that. But again, since everything's happening with RNA in bacteria, it can immediately get translated into a protein really quick. With eukaryotes, you not only have to, splice introns out, you have to find the correct piece of DNA, things like that. You actually have to export that out of the nucleus in a lot of cases for the protein here to get to the ribosomes and actually have a functional protein. So let's take a look at some of the RNA features that keep it, a very dynamic molecule, while keeping in mind, again, the major question of this lecture, which is, does RNA or DNA represent the genome or the state of an organism better? Or maybe does it represent the population of organisms better? Okay, so looking at RNA, we've covered these informally because I really wanted people to know what RNA, why it's a transient molecule. Remember, RNAs have those really, they have tons of those OH groups. They have twice as many as DNA, remember? So it has 2 ends of protection, one on the 5', one on the 3'. On the 5', there is an enzyme that will add a nice methyl cap. This methyl cap is great because it's very unreactive. Nothing's going to be able to chew from this end very easily. Perfect. The other side is the poly A tail, and we've talked a lot about this, and this was actually kind of on the exam and everything. At the end of an RNA transcript, you often add a massive amount of A's, and then you have a small little PAP cap right here, but it synthesizes just tons of these adenes and adenosines because your RNA is going to start to be chewed on from this 3' end. That's just the natural mechanism. We'll see one of the targeted mechanisms of that. This is basically how long this tail is basically how long this RNA has to live, unfortunately. Well, kind of a morbid example, but the longer the tail, the longer this RNA is going to last and make more copies of itself. The shorter the tail, the more transient this gene needs to be on. And that's a very good way, for example, to control how many copies of the protein actually show up. Because remember, gene dosage comes right down to how many proteins do you have active running around the cell doing their job. Okay, so poly-A tail is a great way to control that amount. Okay, this is just a basic slide. You have DNA, this is going to have a promoter, this is going to have genes. These genes are made in eukaryotes of exons and introns. So this is your pre-RNA, this is unprocessed, and remember that you splice the introns out in a lot of cases of RNAs. So you see how these red introns, they're gone. They get spliced out because these are non-coding regions. Now we've talked a lot about why introns are important. One is that sometimes these can be those enhancer sites. Remember the pieces of DNA that go and activate? Those can be introns in the genes, so that's really cool. The other is that in different cells, so that's the thyroid and the brain, you have the same DNA, but because you can select which exons, so remember the coding side, which exons are going to make it, so in this case, in the thyroid, you're going to have the 4 calcitonin, basically the 4 calcitonin exons, they're going to make a calcitonin protein isoform. So this is all coming from the same DNA. If you include all 4 exons, or at least in this first order, this is the protein that you're going to get from this DNA gene. However, you can have an alternative splicing model where you include 5 and 6 instead of 4. See, there's no 4 in this one. That means you're going to end up with CGRP, something completely different. And although related to calcitonin, this is a protein isoform. It is a different protein. It has similar regions. See, it shares 1, 2, and 3 regions. But again, like we've talked about with protein folding, the interactions that those 1, 2, and 3 regions are going to have are likely going to be pretty different from those that they interact with over here because of the addition of 5 and 6 over here. So this is how 20,000 genes in a human can create 150,000 possible proteins. And this was a huge problem in science for a long time, but we figured out, hey, you can switch around and choose which exons you want to actually put into your gene. And so it's very cool, and this complicates things, but it's a very cool dynamic process of how DNA is a blueprint. It's very comprehensive, but it's the decision between what RNA you make that is going to really cause different big differences between cell types and genes and things of that sort. So very cool, very important, and controlling this process can have really interesting effects. We saw in the Journal Club with the Merle phenotypes, just controlling the region of one exon via splicing enzymes can create new organisms and, new phenotypes at least. Okay, how do you splice out an exon? This is very dynamic. We've talked about this once before. Exons will oftentimes come complete with regions that will bulge, basically. They come complete with DNA sections that are splice sites. You need 2 on each side. Usually there is a consensus to these. At these points and at this branch point bulge right here, you can cause nicks, and two of these can kind of react with each other and split each other and turn into what we call a lariat. And it's going to split off this exon right here and split. This RNA is going to get reannealed nicely together. Okay. So in this case, some introns can do this completely by themselves. The DNA, the RNA in this case is reactive enough, and it has a cool 3D shape enough that these 2 sites are going to actually interact and split each other and cause a chemical reaction to remove the intron itself. Oftentimes, though, you will need a protein or a protein complex called a spliceosome or spliceosome complex. These are proteins that exist to find these splice sites, basically pinch these 2 together, and then cut out the introns. Okay. So this is that full process. You have to initiate everything. You start elongating. And then at this point, the 5' cap, remember, because you're starting at 5' and you're going to 3' this direction, you start at the 5' cap. This caps the end of the RNA so that it doesn't start immediately getting degraded right here. You have introns during this time that are going to basically pinch and then split. And then at the end, you have polyadenylation factors and other enzymes that are going to add that nice poly A tail. And you're also going to have like a stop sequence that's going to make either that helicase bind or a hairpin is going to form. And then eventually you end up with this nice RNA right here. So a lot of the splicing can actually happen while this is happening. That's how, that's how intricate this process is. And it does need to be very efficient. So very cool. You can see these little splicing factors. I should have pointed this out. These are what can bring these ends of these introns together and then cut them out. So introns can do it themselves sometimes, but oftentimes they need protein help right here. So losing these is a big disaster because you start including introns. Okay, another thing with eukaryotes, you have to decide if an RNA stays in the nucleus or goes outside the nucleus. So in this case, the blue is the nucleus, cytoplasm is out. So let's say you have a nice new fresh RNA, you need to actually bind that to something called exportin and RAN. These are two partner proteins, they bind the RNA, they send it out. Then this is going to dissociate and you have nice free RNA. Good job, you made it basically. This is a whole nother step of regulation because you don't just get to tag anything, and the RNAs need to have sites that come complete to bind this and allow them to be exited. So remember, the nucleus is kind of like a NORAD. It is the command center, it's everything. You don't want stuff getting in. What is something that you do not want getting in your nucleus? What is the genetic material? What is something that inserts genetic material that you do not want in your nucleus? It's a video, so usually I give you 8 seconds to think about this, but pause if you want. But just like we're encountering right now, viral genetic material. All of this process and why eukaryotes are so well adapted against viral infection in a lot of cases is that they have an entire physical barrier here to keep viral genetic material out. Genetic material in a lot of cases to get in here has to fool our cells and basically get itself imported in this case. So viral genetic material in a lot of cases has to get itself tagged with something that is going to bind importin and RAN in this case and allow it to go in. In a lot of cases, the RNA that comes from something like an RNA virus, like a coronavirus, well, it just exists in the cytoplasm and it's just going to get translated right out here. It doesn't necessarily always need to go back in. So that's why RNA viruses, while a little more delicate, they can just start translating the minute they get in the cell. That's why coronavirus is so difficult, is that it just turns your proteins, your ribosomes, into a printer and makes viral proteins. So not a great deal for us. But under normal circumstances, again, with viruses, the nucleus provides a great defense against just allowing in genetic material. Okay. The other thing, so remember we talked about mRNAs, they have a half-life timer. Not only because they're getting degraded just naturally by some other stuff, but we have stuff in our cytoplasm that degrades RNAs. And this plays into the half-life. As you can see, this is spitting out little RNA nucleotides. These get recycled to become RNAs someday again. So that's part of the process. But the real part, there's 2 real reasons behind this. So on the chalkboard, I'd write 1 and 2. One is that RNAs need to be transient. You can't have RNAs just producing their proteins a million times over. The other thing is that these kill viruses for us. These go around, they find viral material, and they chew it up, okay? Because otherwise we would just have viruses putting RNA in, and it would just start getting translated. So only viruses that can fool an exosome, or at least put in so much RNA that the exosomes can't handle it, they're the ones that are going to get translated and cause infections. But the idea is that there is something like this, and there are other versions of exosomes that exist that chew up genetic material that's not ours. Our immune system has a lot of innate tools that see RNA or single-stranded DNA, stuff that does not belong in a eukaryotic cell, and they start blowing everything up. Again, I've talked about how powerful your immune system is. It is very good, but it needs help inside the cell too. So these are very cool examples of your cellular defenses. And it's kind of what I want you to address in the discussion post, which is these viruses are things that we've lived with and against. And by we, I mean just eukaryotic cells for a long time. So all of the machinery that we see in our genetics, things like that, it's this really strange partnership of defending against foreign viruses, but also co-opting the machinery that they sometimes bring in to utilize and, allow diversity in a eukaryotic cell, like something through crossing over, for example. So this is really cool. Keep that dichotomy and that fight in mind that eukaryotic cells have against viruses, and also really keep in mind what viruses really are in that case, Okay, one last thing, and this is kind of hypothesis territory. This looks like a galaxy, but what this is a collection of non-coding RNAs or RNAs that have been chosen not to code for something. This is a P-body, or processing body. Right now, what we know is that RNA that is not destined to make it as a protein, they'll start to congregate in these bodies, basically. And you can see these in your nucleus, and they're these faint, small little dots. We're not 100% sure why certain RNA collects here and other portions of it don't. To me, this is something really fascinating, and again, kind of one of these black boxes where if we figure out maybe why the rhyme and reason of this is, it'd be really interesting. For me, a guess would be that these are a way not only to remove, amounts of mRNA from the genome that, we don't need to code these anymore. It's also a lot of energy to open up and make RNA, but sometimes you don't want that RNA on in the cell. This is a way to remove that, or, manipulate dosages. Perhaps this is an innate defense against genetic material that shows up that's not from the cell. Again, a viral defense mechanism. I'm not 100% sure. It's a really cool, kind of a cool little unknown thing. I know that we, aren't going to strictly test on stuff like this, but something you may encounter in the future. Awesome. Okay, so with all that, this is something that you're going to address in that lab 4, because this is how a lot the disease that I study is defined. Currently is defined by RNA. Is it better to use DNA in this case? Are DNA alterations that cause large pathway effects and downstream signaling issues and mutations, mutated proteins, is that the big key factor to define? Or is something like RNA that shows at this moment, this is what the cell looks like? At this moment, these are the genes that the cell is expressing, and it's much more reactive, things like that. So again, these are kind of just the ending questions. I kind of addressed this just now. And in this case, both of these kind of play into this mega question up here. Of these two molecules, which one is king? Which is the one that is going to lead us into a better understanding of not only life itself, but the human genome, what makes us who we are, what makes an individual person who they are comparatively, let's say even to an identical twin, because it's obviously not DNA in that case. But can we really say it's RNA either? And another answer here is that the scope of this question is too small, that DNA and RNA are part of the same kind of equation that we aren't considering, especially when you look at human evolution, that human evolution takes on a new role as a biocultural phenomena, and that maybe humans kind of break the idea here. So again, very cool stuff. Keep viruses in mind and what they do and how the machinery in our cells that gives us a lot of that power that we have, how that kind of comes from a viral origin. So think about that. There's another question that's purely more on viruses, which is interesting. And also consider how many defenses we have against viral RNA and DNA showing up in our cells, and why that kind of limits our genetics in that case, like we kind of just talked about. Okay, awesome. I will see you this week.